There are many factors that go into gauging the success of a cell sort. In today's presentation, we're going to talk about the 5 pillars of cell sorting: viability, recovery, efficiency, purity, and proliferation. The agenda today is presented on this slide. We're first going to dive into a brief introduction into the cell sorting process, then we'll transition into the 5 pillars. So if you're an experienced cell sorter, please bear with me for the next couple of slides because I want to take a couple minutes to go over some of the basics. Cytometry is a term that describes the physical or chemical measurement of a cell. Flow cytometry is a technology that was developed in the mid-1960s and provides insight onto the physical and chemical properties of these individual cells. Each cellular measurement is aggregated to provide the distribution of measured characteristics of the sample. And then if we take that a step further, cell sorting is an extension of flow cytometry. Cell sorting allows the user to decide on which populations to be sorted. There's a wide variety of sorters on the market today, but today we're going to be discussing droplet-based electrostatic sorting. This is where the droplets are collected in a wide variety of collection plates, tubes, and slides. So we're going to cover all the pieces that go into sorting. And these are the 7 pieces shown on this slide. And so it all starts with the hardware. There's flow cells, lasers, nozzles, optics. We're not going to go too much into the lasers and optics portion, but we're going to go more into how droplets are deflected, how to collect them into the media, and all of the other components that go into the hardware. Then we have the detection side, where we have the cell that passes through the laser, and then we get to set our gates and be able to sort from specific populations. Making droplets is one of the most critical portions of cell sorting. So we're going to talk about making droplets, how to properly calculate the laser delays, how to charge the stream, how to deflect the droplets, and then lastly, how we can officially get our cells down to downstream applications through collecting them in different media types. So as far as the basics go, certain cells can be more fragile. And so we basically have a diagram on the right here where the cells are injected into the stream, and which the sheath comes around, hydrodynamically focuses our sample. We vibrate the nozzle and generate drops. Then we pass a laser through the stream, and then we measure the scattered and fluorescent light. And then we have our break off, which is a little bit lower. And then we electrostatically deflect these using high voltage plates. And then we can collect these into tubes. So keep in mind that we're actually deflecting and charging the drops. We're not actually deflecting and charging the cell. The cell lives within the drop. And so it follows the drop into the collection tube. And certain cells can be more fragile. And so selecting the appropriate nozzle size, sheath pressure, and boost settings can really give you an advantage to optimizing your sort before it even starts. Make sure the instrument has been properly maintained. And so there's lots of different parts that interact with the cell, and that's the sheath, the actual, the drop. And so having things like the sample line, making sure that the sorter is in good maintained order, will really give you a good head start into getting off to a good start when sorting. Make sure to set the desired temperature for the sample well in advance of the sort, just to let the system kind of reach that temperature and stabilize. And then sample prep is critical and often overlooked. Make sure to use proper washing steps and making sure that the sorting and collection media that the cells are going to be in are optimal for that specific type of cell. And so once you're done sorting, sorting is oftentimes just a middle step in the process of your experiment. And so what can you do after sorting? Well, this is where you can kind of resort or reanalyze your cells to ensure that you received good purity, good viability, and some of the things that we're going to talk about. So you can actually just rerun those cells or you can actually culture them or use them in other downstream applications. So looking at the basics a little bit deeper, that we can actually look at some of how these pieces of hardware work. And so in the middle of the screen is the actual nozzle body assembly. And that's where it's a fluidic assembly where the sheath comes in up in the top right part here. And it comes around and gets injected into the actual nozzle itself. And then we have our sample, which is introduced through the injection needle. And as soon as you run your sample, that's going to hydrodynamically focus. And so hydrodynamically focus is what gives you a nice core stream. And so when the nozzle is vibrated, it's going to produce drops. Everything is going to flow smoothly down through the orifice. And once it's vibrating, then you'll form drops. And so it's really important to have a nice core stream that's going to give you nice and tight CVs. That's going to allow you to generate these droplets at different sizes. So we have 70 microns all the way up to 200 micron tips. And so that's going to give you a wide variety of droplet sizes, which can be optimized for your particular experiment. On the right here, we have the Invitrogen Bigfoot nozzle chamber. And so this yellow line is the actual sample line that goes up into the injection needle that basically delivers your sample into the nozzle. And this is actually in the unclog station. And when it's fully retracted, it goes into the sorter where the lasers are located below into this area, which is the laser interrogation chamber. So one of the most important parts of the nozzle assembly is to generate droplets and make them stable and predictable. And we typically use a piezoelectric crystal to do this. What that crystal will do is vibrate up and down and produce a very consistent droplet. And you can actually change the breakoff height based on the voltage applied to that crystal. And that's just going to be basically an energetic wave that's produced. And so you can see on the far right, this is a strobe image with an LED light that illuminates the droplets. And then we have a lot of machine vision in there to kind of analyze this image. So we can get things like the neck width, which is 15 pixels wide. We can get things like the length of the satellite and the overall gap length. And so all of this will allow us to predictably control and make sure that we're always maintaining these droplets at the same spot. So that will make things like your drop delay much more accurate. And all of this is done automated through the system. And so there's really no user interaction once you get maintaining on a specific point. You can always go back and look at different frequencies and amplitudes, but typically day to day, these are going to be very consistent at the same frequency, same amplitude with the same nozzle tip. So as a cell or particle passes through the laser, we can measure the properties of the cell. If the final gate of interest is accepted by the sort criteria, it will charge the stream at that precise moment that the cells enter the last attached drop. And so that's where we can basically sort from different gates, and then we can neutralize the stream, which is called the fanning, and then wait for that next cell to come through. And so all this is related back to the drop delay, maintaining a very stable breakoff point, and then being able to deflect those cells into the collection tubes. And the stability really buys you things like sort stream stability. So your side streams, when you're doing a six-way sort, you want really stable, consistent breakoff point. And so that could be looking at the height of the breakoff and really just trying to get those side streams to be very stable. And so you can reduce what's called fanning and spraying. And so you can get a nice, consistent deposition of your droplet, which contains your cell of interest, into your collection tube. A lot of questions typically come up on a jet and air system versus a cuvette-based system. This slide highlights some of those differences. A jet and air system uses a stream that's jetted through a nozzle directly to achieve a very consistent stream velocity. So here we're maintaining 30 meters per second consistently. A cuvette-based sorter is going to have a cuvette where the sample passes through, and that's going to change the velocity of those particles. And so in this case, it might slow down to around 6 meters per second, then reaccelerate when it's hydrodynamically focused up to 10 meters per second, 20 meters per second, and then finally reaches its top speed of 30 meters per second as it exits the orifice of that nozzle tip. And so the biggest difference really comes down to pulse width. And so a jet and air sorter, because you're traveling at that faster speed and consistent, you can really achieve narrow pulse widths. And so in this case, we're under 1 microsecond pulse width. And so that's basically the time that that cell is going to pass through the interrogation laser, and then you're going to collect that on your, say, PMT or some sort of photo detector. And so I like to relate this back to microscopy, and that shorter pulse width is going to allow you to look at the next cell coming through quicker. You're not going to have as much overlap. And so you're basically getting more and more resolution, so you can see more as your sample comes through. And that's one of the real advantages of having the jet and air sorter versus a cuvette-based sorter. And that ultimately comes down to throughput and seeing more of your cells as they come through the system. So kind of looking at some of the automation features, and one of the most critical is drop delay. And drop delay is the time from the laser interrogation point to the break off. And so we need to get a very good and accurate measurement. And there are many ways to estimate the drop delay. Some systems use microscope slides, some have automated system. The Bigfoot actually has a mini cytometer built into the system that measures the drop delay in a very accurate manner. And this allows you to achieve a high quality cell sort because you're going to get higher recovery. And so making sure that the drop that you sort, that you deflect, actually contains the cell. And we'll go into some more details on how drop delay can influence certain parameters like recovery more than, say, purity. But this is all automated. This is an example on the right of the Bigfoot automated drop delay plot. And so on the bottom axis, you have droplets, so 26 drops to 30 drops. And then you actually have an intensity. And so once that the drop delay is accurately met, it's going to deflect drops with actual beads in it. So these are 10.6 micron beads, and that's going to give you an intensity of, in this case, maximum intensity of 3,500. And so that's basically saying at that point, so in this case, 28.06 drops is the exact point at which the most cells or beads in this case are going to be measured by this cytometer inside the system. And so you can use that drop delay for your cell experiments and bead experiments, and that will ultimately give you the most accurate drop delay. And so looking at taking that a step further, we have a newer feature called cell-based drop delay confirmation. And so here, we always recommend doing drop delay confirmation on a slide just to look at, say, a drop before the drop that was picked, the main drop delay, and then one drop after. And you should see, say, almost 100%, maybe just one or two in the drop before or the drop after. And you can actually plug these numbers into SQ, and that will readjust the drop delay for you. But these are typically drop delays done with 10.6 micron beads, and cells as we know, range in size and can be either larger or smaller. And so it's always good to do what's called a cell-based drop delay. And so you can actually use the gate of interest that you're going to be doing for your sorting to sort onto a slide, and just make sure that the actual drop delay that was measured is going to be best for your particular cell's interest. So this is just looking at PBMC that were CD8s with FITI, and so we can verify that all the cells were falling within the correct drop delay as measured on the system. And so machine vision is one of the biggest strengths of Bigfoot and allowing a successful self-store, because it automates many of these processes for you, whether it's setting up the drops itself and kind of looking for the best point to maintain. And so you don't have to worry about, say, satellite length to gap ratios. All of that is done for you just by clicking the maintain button. Things like alignment of the nozzle, that is all done for you. Setting up the side streams. So when you're looking at, say, a 96 well played stream setup, that's going to go through and it's going to pick the best charge phase. It's going to pick the best of fanning. And that ultimately is going to give you the best side streams. So that's going to reduce things like fanning. And so all of this is done through using 6 cameras. These are the CMOS cameras to automatically help facilitate the setup of the system. And ultimately, this will allow you to have a better sort, because all these parameters are going to be optimized during the automation, whether that's a 1-button QC, which does everything in steps that are sequential, or if you want to go through and do these manually, there's also a new mode for power users, and so you can actually look at doing your own maintaining of droplets. You can do your custom drop delays. All of this is flexible, and so you have the options of either letting the computer do it for you, walk away, get a cup of coffee, come back in 10 minutes, or you can just allow the manual operation, which can get you there a little bit quicker if you want to do it yourself. So lots of options there. So the first pillar is sort purity. And sort purity is defined as the percentage of the target population and the sorted sample. And so there are different modes, and we're not going to talk about too many of the different modes, such as single mode, which is typically used in plate sorting. That's going to have a more stringent criteria for the position of the cell in the drop, and that typically is going to result in the highest purity. For bulk sorting, typically what we use is something called purity mode, and that by definition just can't have a contaminant cell that is to be probably your best balance of efficiency, speed, and recovery. And so we can modify all these sort modes with a custom setting, and so you can actually increase, decrease the sort criterias. But purity is going to be something that you can remeasure after your sort to confirm that you have a high purity. But oftentimes purity is not going to be as impacted when drop delay is off. So that's typically not where you want to go for your first troubleshooting. But you want to look at things like cell preparation. If you have a lot of debris, if you have a lot of clumps, that can contaminate the sort population. That's typically a first thing to look at. Things like concentration, where you have too many sort conflicts, you're gonna look at something like maybe you want to dilute your sample, change the flow rates, things like that can help optimize your purity for particular types of cells. look at things like, so on drop delay, this is a very interesting chart on the bottom left here, where you have theoretical purity and theoretical recovery. And so you can actually see from the blue curve that your purity is going to maintain pretty high, even though your drop delay error is off. But when looking at theoretical recovery, that's going to have a much steeper drop off. And so this chart is basically showing that if you're slightly off in drop delay, that can certainly lead to reduced purity, but it's actually, you can be off by a decent amount and still maintain a decent purity. And so in addition to that, looking at things like break off, stability, and things like gating errors, how you set your threshold, that will allow you to more or less debris. That can also impact purity. And then dim positives that can appear negative after sort due to gating. These are all things that you can look at when you want to try and troubleshoot purity of your sample. So we do a lot of testing on purity, oftentimes with samples like human PBMCs. These are obviously samples that you can stain, you can sort different populations, whether that's a 2-way, 4-way, 6-way, sort into different types of media, whether that's 5 ml tubes, plates. So in this case, for purity, we typically do more bulk sorting so we can get some more statistics. This sample was measured in 2 cases on a 70 micron tip and a 100 micron tip to look at the purity, and that's typically done in the post-sorted sample. So we'll sort these, say like 100,000 or even more, 500, even a million, and then we'll wash the sample line. Carryover, we test on Bigfoot, and it's a standard wash typically gets you into a very acceptable criteria, but you always want to wash your sample path in between measuring or sorting your sample and then reanalyzing, because if you have a small amount of carryover, that could be the difference between 99.2 and 99.7. Is just that 0.05 is going to be the carryover that was in the sample line. So good wash between samples and then reanalyze and do replicates so you can get some more statistics on post-sort purity. And when we collect this, we also measure some of the more important statistics as well. This just kind of is a summary table from the results on the previous slide. The purities typically average out 98% or greater, and we hold this, we actually run this test in manufacturing and integration as well. Typically average out to be over 98% while maintaining a high efficiency throughout for each population. In this case, we used the live dead fixed aqua, which was a viability dye, and that is good to note that it limits the amount of non-specific binding of dead cells, which can also decrease purity. So it's always good to have a viability dye there. In this case, we do have a mode that is recovery enhanced mode. So in this case, in this experiment, we did not use it because we were more focused on purity, but that can also be used to increase recovery. So we also ran the same sample. The sample concentration in this case was about 10 million cells per mil. In this case is showing the 100 micron tip, which for this particular sample, you could use the 70 or the 100 micron tip. In this case, purities, you're going to have a little bit of a bigger drop, but the purities are still around 97%, and we did have good efficiency as well. Both of these experiments use purity mode, which is going to be the most common for this type of bulk sorting when we sort this many cells. So the second pillar is sort recovery. And so sort recovery is the number of post-sorted sample in your tube. So you can actually measure the amount of cells that were in the tube after the sort. And so let's say one example is you sorted 10 cells, or in, maybe say 100,000. And then you go back and you reanalyze your tube and you count 9. So you got 9 out of 10, or you got 90,000 out of 100,000. That would give you a recovery of 90%. And there are various reasons that recovery can be lower than expected. And the first source is drop delay. And so going back to drop delay confirmation, making sure your drop delay confirmation, not only you're sorting, say, if you sort, let's say, 25 beads into your slide, you typically want to count those beads and just make sure that you're getting a good amount of those beads. And so your drop delay is very well calculated. Other areas that you can look into is the cells sticking to the tubes. In tubes, there are various types of tubes. Tubes like polypropylene that might get you a little bit better if the droplet hits the side of the tube. It can slide down. The cell can be happy in the media or the collection buffer. Cells can die during the sorts. You want to make sure that you're using the appropriate sheath pressure, nozzle size, using viability dyes, things like that. Static buildup, the cells, if you do a very long sort, you can actually get some static buildup on the actual tube, say a polystyrene tube. The cells can actually be repelled, so they'll actually jump out of the tube. And then ultimately on a majority of application, I like to say 70 to 90. If you're using, say, a sample that has low viability, very delicate, fragile cells with a lot of debris, a very tough sort, yeah, you're going to be down in that 70% range. But very good sample preparation can help with that. Washing, getting rid of some of the debris, using the appropriate optimization of temperatures, things of that nature, really good side streams, limits the amount of fanning of the side streams. That can really get you much better recovery. And so we often get questions about how do we measure recovery, and that can be one of the hardest or the most difficult things with recovery. And here are 4 different techniques that we typically use. You could use a flow cytometer, whether something that's volumetric, like a volumetric analyzer, like the Attune NXT. That's pretty common. And what you do here is you just weigh the tube to get a volume of the fluid in the tube, and then you run, say, 100 microliters of your sample, and then you reweigh your tubes. So you know how much volume that you have, and then you basically capture your gate. That gate tells you how many cells that you have per microliter, and then you just multiply that amount by the total volume of that was in your tube originally. And so that's going to basically give you a total cell count. And that can work for smaller amounts of cells, larger amounts of cells. It's a pretty robust method. Hemocytometers are also very good, or a cell counter. Typically there you have to sort a little bit more cells. So if you're worried about, if you don't want to have to sort 100 or 200,000 cells, or even less than that, because you want to save it for your downstream application, typically using a cytometer for reanalysis can help with that. But hemocytometers and cell counters are really good at that. Counting beads, you can actually add these to a cytometer. You can add these to something like the Bigfoot, and then get a ratio of the exact amount of counting beads to cells. Then you can basically determine your final cell count or concentration that way. And typically, you can even use the Bigfoot cell sorter to do that. Um, the challenge with using a non-volumetric measurement is that it doesn't account for things like the boost, or if you run through your tube, um, you may have a little bit of liquid in the bottom. So recovery on Bigfoot and other sorters can be a little bit more challenging. So I typically recommend using a volumetric analyzer or a cell counter or cytometer, but that is an option if you don't have any of these other options in your lab. Recovery and plates. And so PBMCs and plates, lyophilized PBMCs, we sort into 96 well plates, typically do one or single cell, and then we visualize these on a microscope. So we have an EVOS microscope that we can use to go back and count. So if we have a 96 well plate, we want 93 out of 96 to have 5 per well. Or if we do single cells, we want to look at each individual well and ensure that 96 of those wells have a single cell located in them, and the fluorescence just allows us to visually see these on the microscope that much easier. In this case, we move from cells to beads, and so we test, from beads to cells. And so in this case, we're looking at 96, 384, 1536 well plates, and then we measure the overall number of wells that have the correct number. We do multi-way, so multiple streams per sort, or straight down, which is a single straight down sort. And so we can look at that into 96, and in this case, the data on the right is 384 well plates. Looking at jerkats that were sorted on the 70 micron tip with very good amount of cells, the correct number of cells per well for 384 in both straight down and multi-way. The top data is 100 micron tip data using NIH 3T3s. I wanted to mention RMAX. It's a very good technique for measuring recovery without having to bring your cells to, say, another analyzer or using beads. And this was termed RMAX and developed by Rui Gardner and coworkers. Very good reference paper in the bottom right-hand corner. And what RMAX can indicate is how much of your sort sample is lost in the unsorted fraction. So in this case, that would be the waste. And so you can actually collect that, rerun that, and look at how many of those cells or beads were actually in the waste when they should have been deflected. They should have ended up in your sort tube. And so that low Rmax value can indicate that there is an issue with the sorter's performance, and that may not show up when looking at, say, the purity of the sample. And so this is just troubleshooting issues related to recovery, and you can go back and look at the drop delay, you can look at sample prep, other areas that could be the root cause of having a low Rmax. So we actually implement this in our manufacturing final integration testing of Bigfoot to ensure that not only the purity is good, but the recovery of the system is good as well. And this is just kind of going into how we measure it using an example in a Bigfoot SQ protocol that we run in manufacturing. We use two different types or two different comp beads. And then we take the ratio of the sorted beads versus the target beads in the waste stream. And so we can get some statistics, and we can get some Rmax values from system to system, and relate that to making sure everything on the system is set up properly to ensure that we're getting a good recovery. The third pillar is sort efficiency. So sorting efficiency is the number of target cells sorted by the instrument as the percentage of the number of target cells detected. And so that is looking at, basically how we can get a good efficiency by the sort criteria. And so if the sort criteria says that if you have two cells that are too close together, you have to have what's called an abort. There's something called a hard abort, and that is um, uh, when a system sees a event that is too close together for the instrument to process, and so it's going to ignore that. And so um, that's the difference between a abort, which is something that physically the system sees and says, "Oh, I can't sort that because it doesn't meet the sort criteria." Whether that's something that you set up on the system or maybe something that the system decides. But a hard abort is going to be different than that, and that's where the system is just going to ignore that, and it's not going to actually include that in the sort processing or the FCS data. And so that's one advantage of the Bigfoot is that it does not have hard aborts. And so it was designed with electronics that handle event rates and processing capabilities well above any experimental requirements. So there are no hard aborts. And so this all goes back to a relationship with the pulse width and the time that it takes for a cell to pass through the laser illumination. And Bigfoot has a tool that you can look at the signals coming through, basically an oscilloscope, and you can actually look at the event windows. So the event windows are on the top here, and so that is going to show how the system is processing that individual signal, in this case, coming from the PMT located off of the forward scatter detector. And so we can look at what that sort decision or what that signal is going to be and how closely those events are spaced out. And so if you have a wider pulse width, there's more likely to be multiple cells. And so having that very narrow pulse width allows you to process these events much quicker, and you'll have more separation in that signal, and so you can actually detect that. And again, that kind of relates back to having a higher resolution camera or a microscope, and so we can go through and we don't ignore any of these events coming through, but rather we process these, and that's called dynamic window processing. And so that's going to give us that ability to process the 1.5 microseconds on a jet nerve sorter, which is going to be much lower than say the equivalent on a cuvette-based system. So 3.3 microseconds pulse width on a 40 micron cell versus 9.9 on a 40 micron cell on a cuvette-based system. So we have much more capacity to look at these narrower pulse widths and process these cells in a much quicker manner to get a little bit higher throughput there. And so some systems on the market process events that are overlapped differently. And so on this example, two pulses are shown to represent the forward scatter PMT signal as the cell passes through the interrogation point. And some systems will just discard these events. They won't include them in the FCS or the sorting statistics. And this can be changed in settings, such as varying the window extension. So the window extension is how much additional time the sorter processes the event. And so if you have a larger, and these are typically microseconds, so if you have a larger window extension, you can basically take longer to process. And if you have, if you're running at a higher concentration, a higher sort rate, you can have a lot of events coming through at the same time. So if you just vary that window extension, you can actually just start to, on other systems, ignore a cuvette-based system. Some cuvette-based systems, you can just start to ignore these events, and they don't fall into your efficiency calculation. And this is just an example of that. So where you have the window extension and how it impacts the reported statistics, going from a window extension of 0.5 microseconds up to 25 microseconds, you can just see the increase in efficiency. And that's basically because you're extending the amount of time that it's processing. And as events get closer and closer, uh, it's going to just start throwing those events away and not processing them. So it doesn't really hurt your, it really just gives you a better efficiency because you're just having more harder boards in this case. And so that's where making a true volumetric head-to-head very comparative for efficiency to really kind of make that statistic transferable across multiple different types of sorters. Bigfoot has the ability to change the window based on the dynamic algorithm that it uses. And so on some instruments, there's a fixed time period, and that is going to be something that you can change, but you can't change it dynamically. Bigfoot will actually change that dynamically. So when you have two cells or particles that are close together, if you do have a window extension overlap, then it's basically going to count it as a doublet. And so you're going to start to see that signal on a Bigfoot. So you're going to have a higher event rate because you're actually detecting more and more of these particles. This is an example of if there is an overlap, how the Bigfoot would process that. And it's going to split that signal because it does have the dynamic processing. And what allows us to be able to process that much faster comes down to two things. One is the fluidic stability from the jet and air system. The other is the FPGA architecture that was designed to accommodate these speeds. And so we can go at a much higher rate than any, say, event rate per second that would be possible. So I think the board is spec'd to over 600,000 events per second, which physically is not possible at these pressures and speeds. So it was designed with that in mind to be able to process these events. So in this example, Bigfoot would process all 4 events, while other sorters would process just 3 events. And so that may not seem like a lot, 3 out of 4, but when you're processing millions and millions of events, that just allows you to finish your sorting much faster and not use as much of your precious sample. So here the window extensions, theoretical examples of a fixed window extension where it sees 5 total events and it only processes 3. And so this would be representative of some cuvette-based sorters where it's going to process 3 events, it's going to sort 3 events, but it's going to have to abort 2 because they're too close. However, it's going to report 100% efficiency because of the hard aborts. With a dynamic window extension on the Bigfoot, we're processing 5 events. We're sorting 3 because 1 doesn't meet the sort criteria because you have a contaminant that follows a positive. But then you have your electronic aborts are 0 because it doesn't have electronic aborts. But it does have 1 true abort because of the purity mask, which basically says it can't have a contaminant in the drop. And then your efficiency reported is 75. So you're actually getting the true representation of the efficiency of the system. And so we have to go back and model this, and we can use Poisson statistics or Poisson distribution as described in the Linmo paper. And that is ultimately looking at verifying what our reported efficiency is versus the actual efficiency. So we can do this with beads, and we vary the percent of the total target population in the final gate. And we can correlate that back to efficiency and get a good model of the change in theoretical efficiency versus observed or actual efficiency measured on the system. And the Lemmo paper references is at the bottom here, which is where we can go and calculate the theoretical efficiency that we should expect from the system. Observed versus predicted. So this is just looking at different modes. So previously shown was looking at purity, we can also extend that to look at single mode and rich mode, and we can see good correlation between predicted and observed across the different modes as well. Droplet spacing and polarity flipping. Droplet spacing is a really good technique for improving recovery, but it will have an impact in efficiency, and that's why I included it in this section, because the actual efficiency will go down because you're telling the system that you don't want to sort if your target sort decisions are too close together. And so you can set that between 2 drops, 4 drops, 8 drops. And you can actually see in the image above, when you have your drop spacing of 0, which means basically the drops can be right next to each other, that's going to induce more fanning in the sort tube, and that's going to lower your recovery. But if you have it on, it's actually going to impact your efficiency, because you're basically going to abort if it does fall too close together. And so you can have lower efficiency, but you can actually increase your recovery. And so it can be a trade-off that you make, and you can actually do this live in SQ to make that decision on drop spacing. But ultimately, at the end of the day, recovery is going to be a very important aspect. And so you can adjust the drop spacing during your sort. The other is polarity flip, which is a feature that will actually reverse polarity on the high voltage plates. And so that's actually going to flip from positive to minus. So if you have a positive charge built up on your tube from sorting for a long period of time, that's where you start to see the cells jump in and out, or jump out, just because they're the same charge as what's built up on the tube. We can actually polarity flip at a certain amount of volume, which is going to correlate to time based on the number of drops going in the tube, and that will actually flip from positive to negative. So that will minimize the amount of cells that are jumping out of the tube, but that can also affect efficiency very slightly. It only takes less than a second to actually for the plate charge to change, but during that time we'll just abort those because we want to maximize recovery. So these are some decisions that you can make during your sort, or even before your sort, to maximize recovery, but they will have an impact on the reported efficiency. We can actually look at event arrival statistics, and this is a newer area that we're looking into, because Bigfoot has the precision power to track individual drops with 8 bits of precision. So each drop can be broken up into 256 different individual slices. And so we can actually look at the event gaps per drop. We can create a more or less a statistic that looks at what we term fast events. And so the Poisson statistics for efficiency do not factor in sample prep. And so if you have clumpy, more aggregates that are going to produce, say, more doublets, triplets, etc., we can actually look at the arrival times for beads and for cells, and then look how that impacts efficiency. So if you have beads, you're typically going to have less than 1%, or in this case 0.1%, where fast events, the efficiency was 98%. If you have CHO cells, which are going to be a little more sticky, a little bigger, 0.8% fast events, and that's going to lead to an efficiency of 85%. So looking at these arrival statistics, we can actually go back and look at how that influences, or how it can be improved through sample prep, just to be able to get you better single cell suspensions, so you can more efficiently sort these cells. And then we can also look at improvements, say if you're adding a surfactant or you're not vortexing your sample, what are those effects on efficiency, and improving that single cell suspension, whether that's vortexing or any type of sample improvements. You can actually start to look at the differences between these fast events and the effect on the efficiency. The fourth pillar is sort viability. And so the last two, viability, proliferation, we'll try to get through a little quicker. But post-sort viability is a percentage of live cells in the post-sort tube, and that will differ um, uh, on their ability to survive the sort. And cell preparation, again, can help improve viability by reducing clumping, reducing fanning, things of that nature. And it's very simple to remeasure post-sort viability. We can just reanalyze the sorted cells, typically with a different viability dye, and then we can report the percentage of live to overall number of cells. So if you sort 1,000,000 cells, just rerun those cells, have the same gates, look at how many fall within the live or dead gates, and use any sort of viability dye. In this case, we're sorting jerkats at 100 or at 1,000 per second on 100 micron tip at 30 psi, using Cytox Advanced as our viability dye, and then just rerunning those cells. Then we can look at them under the microscope, try to optimize our sorting. In this case, there's very few dead cells in the post-sort viability. There are some tips on here to help with post-sort viability, whether that's optimizing buffer solutions, checking your media targets, make sure you're landing in your tube, drop delays, temperature controls, pre-sort treatments to improve the single cell suspension. All of these should ultimately help with post-sort viability. And the fifth pillar is post-sort proliferation. So ultimately, we want to look at how these cells are proliferating after the sort. And so we can make sure they're not stressed, they're not damaged, which can prevent accurate and meaningful downstream applications, such as PCR. Peter Lopez and his team looked at cell-induced cell sorting, or stress-induced cell sorting, which is a really good paper that kind of looks at the downstream applications and how they are affected. Some of the data presented here is looking at the cell lines or cells in plates and tubes, making sure they can culture, form colonies, healthy-looking colonies. In this case, on this example, is jerkats that were monitored in colonies in 96 well plates, 15 days post-sort, looking at colony formations across the 96 different wells. And when you combine good recovery results with good cell proliferation, you can have a better chance of success in your downstream applications. This was a similar experiment about GFP positive, where we can image easily with fluorescent microscopes. This is the EVOS microscope with the GFP filter block, so we can monitor the growth rate of these cells on day 0, day 3, day 5 at the same magnification post-sort after 100,000 cells were sorted into a 12-well plate. This is an example where we sorted cells and we had an unsorted fraction. So cultured for 5 days, the unsorted fraction and the sorted fraction, 200,000 cells, culture for 5 days, check for viability and counts. The cell viability and cell count for the sorted cells were equally high in some cases. In these cases, for the unsorted as well as the sorted. And so the stress that the cells experienced from the sorting process did not slow down the proliferation of these jerkat cells. In addition to microscopy, we can use flow cytometry imaging to monitor cell proliferation and look at the quality and health of the cells. In this case, 3, 7, and 10 days post-sort. We can image live dead on the attuned site pics. In this case, the cells looked healthy, so we continued to culture these, and all the way out to day 16, where we started with 1,000. We saw them proliferate into a very high quantity of cells 2 weeks and beyond post-sort. So these were ultimately cultured and had very high viabilities several weeks down the line. In this example, we did adherent cells, so NIH3T3s, sorted into 6 well plates using the 100 micron tip at 30 psi. In this case, we did straight down sorting. These cells proliferated nicely over 14 days, maintained viabilities over 90% over the course of 2 weeks. Recovery and viability are all factors which we use to measure the success of a cell sort. And so when looking at the advantages of the Bigfoot cell sorter, once we achieve these 5 pillars, you're ultimately going to detect more cells. That will give you more information from your tube of cells during your sample analysis. The samples can be run faster, and so this all goes back to pulse width and being able to get more sorts quicker, sort rates, faster sort rates. And you can sort more rare cells because you're seeing more events. You can finish your sorting of those rare events quicker. You can reduce the sample runtime, and that will ultimately increase the cell viability. And so having a higher cell viability because you're taking 30 minutes or 10 minutes as opposed to 20 minutes, or your plate sorting is taking less than a minute, where on another system it takes 3 minutes. So there's not only the throughput of getting done quicker, but you can actually improve your cells and your viabilities by getting through that sort quicker. And we log all the FCS files with individual droplet indexing. So there's a lot of information and a lot of data that you can get statistical distributions from. And so that will enable you to be more confident in the cell sorting process. And then as we talked about, the machine vision and smart algorithms track and help maintain all the system performance. That can ultimately help you train new users with ease and get people more comfortable with the sorter faster. So with this, we've reached all the 5 pillars of the cell sorting. If you have any questions, comments, I would definitely be open to taking those. And I really want to thank you all for your attention today. And I'm going to pass it back to Daria. Thank you.
Thank you everyone for joining us today for the webinar. Reminder, you can still submit questions. I know we're running a little bit over time, but we'll be able to take a few questions now. And questions that we don't get to, we will be able to answer over email because we have your contact information through your registration form. So you will get your question answered even if it's not at this moment. We had a few questions submitted, so I'll start with those. First question, while calculating the percent recovery, the number of cells sorted would mean the total number of cells before sorting began, or is it the sum of total cells collected in the 2 channels post-sorting?
Yes, that's a great question. The denominator in the recovery calculation is the total amount of cells that the sorter sorted or told you that it sorted. So if you put in a sort limit of 100,000 or million, that's ultimately going to be what your denominator is going to be, and then you're going to measure how many cells you actually have in the tube. And so if you get 800,000 out of a million, which is what you ended up in your sort gate and where the sorter stopped, that would give you an 80% recovery. And so ultimately, knowing how many cells that fell within that gate when you stopped your sort, that will give you your denominator, and then you have to measure how many cells are actually in the sort tube.
Great, thank you for that. Next question. Can you discuss droplet spacing tweaks and what it does to potentially benefit efficiency?
Yeah, it's another great question. So droplet spacing is a relatively unique and new feature, and that basically will allow you to optimize your fanning into your tubes. And so if you have cells that are very closely spaced together, they have the same charge, they can repel each other. And so if they're too close, we want to keep our side streams clean, and so we can get very good recovery. And so if these cells or these drops are very close together, those will actually be aborted. And so you can reduce the amount of fanning. And so there are default settings, 2, 4, and 8. Those will actually increase as you get further out, where fanning is going to be more prevalent. And those are just default settings. Those can actually be tweaked in real time while you're sorting. If you notice you have any side stream fanning, and so it's a very flexible and we just like to note that it can impact efficiency, but the trade-off is better recovery. And so it's something that you can optimize for your individual experiments and kind of fine tune based on the cell that you're sorting.
Great. That was a great question. Next question is, does the Bigfoot like faster sample rates over pressures? So for example, over 100%. And along this line, what would be the recommended particle spacing? For example, 1 cell over 5 drops or something else?
Yeah, so Bigfoot can run at a variety of different flow rates. You can kind of select that based on your sample concentration. I would say that from a processing perspective, it can handle very high concentrations. I think the important thing here would be to look at side stream stability as you're sorting, just to make sure that you're not seeing fanning on the tubes. If you do, you can typically maybe dilute your sample, run a little faster, and that will give you a little bit better spacing, in this case, cell spacing. And so that will clean up a lot of the any fanning or side stream instabilities. But that really depends on things like your cell size to nozzle tip size ratios, and so there are different factors in there. But I would say that you can run at various speeds and optimize your concentration, anywhere from low to medium to high on the Bigfoot.
Great. Thank you for that detailed answer. The next question is not about cell sorting, but about the webinar. Someone asked if this webinar will be, the recording will be available, and also commenting that there's so much information and it's a great presentation by Matt. I completely agree. Thank you, Matt. This webinar recording will be available. Visit our Bigfoot resources webpage. So you can go to thermofisher.com/Bigfoot. That will bring you to the general overview page for the Bigfoot, and just select the resources tab, and we have a whole lot of educational videos there. And this video will be posted in 3 to 4 weeks from now. You'll be able to find it online on that webpage. I think that's all the time we have. We ran 5 minutes over. Thank you for everyone that stayed with us. And thank you, Matt, for a wonderful presentation and for answering these questions. Any questions we didn't get to, we will reach out to you by email. Thank you, and have a good day, everyone.